Hey guys, this is Kendall Terry, and this is a lecture on membranes for Bio 2 and AP Biology. We're going to just go through a series of questions really to try to understand membranes. The first being, what do you know about membranes? Well, you probably to this point know that membranes are made up of a phospholipid bilayer. This is a phospholipid right here on the screen. Phosphate head, the lipid tails, fatty acid tails. It has a hydrophilic region that likes water up here, a hydrophobic region, those tails that do not like water down here. And because that molecule actually has both regions, we call it an amphipathic molecule because it has both hydrophobic and hydrophilic. Whenever you get a phospholipid in water, a bunch of them in water, what happens is they create a ball, in essence, where the phosphates will be on the outside, tails will be on the inside. But in your cell, the inside is also made up of a watery material, so it creates this bilayer membrane where you have phosphates on the inside, phosphates on the outside, tails on this membrane inner layer then. So this creates a separation between the outside and inside. We know that cell membrane is selectively permeable, and this is key, meaning it only lets certain things in and out. We're actually going to spend about half of this lecture talking about that. And it's also what we call a fluid mosaic model. So we'll get to the fluid mosaic model in just a minute. What are the four components groups of the cellular membrane? Well, there's the phospholipid bilayer, which we discussed. There's transmembrane proteins. Transmembrane proteins are proteins that will transverse or go the entire width of the membrane and stick out on the inside and stick out on the outside. And those membranes are very important to getting things across that membrane. There's interior membrane protein network, where some proteins are going to go all the way through the cell, and then there's going to be other little proteins that will kind of be right here inside the cell that will attach here. And possibly, depending on the reaction coming from this protein, will cause something to happen down here inside the cell. And there's a network of these proteins inside the cell that are connected up to the cell membrane to cause different reactions to take place. And then there are cell surface markers, and these primarily are made up of carbohydrates, and these will determine or mark what cells are what. And we'll look at some examples of that in just a second as well. So here you've got all of those. So these big globs here are the transmembrane proteins, those proteins that go the whole length or width of the cell. You can see that they stick out on both sides. Then you've got this protein network in here. You can see these little ones hanging out just in here in the middle of the cell, and they're going to be connected and do different things. Here's the carbohydrate markers. We'll talk a little bit more about them in a minute. And, of course, then that phospholipid bilayer. So what makes a membrane what we call the fluid mosaic? Well, it's called that because everything that you see here in this picture can actually move around. Nothing is solid in place where it can't move. There's a lot of protein shifting. There's a lot of moving going on, even in the phospholipids themselves. You'll have a very rare occurrence, but you'll have an inside and outside one flip. You'll have a lot of back and forth movement amongst them as well. I like to think of this picture as kind of like ships on an ocean. So the proteins are ships, and they're floating around on this ocean surface. They can move and change which one is in which place, so on and so forth. It's movable, and that's where it gets its name. When you look at an integral protein, how are they held in place? Because they are stuck inside that membrane. Well, the way they're held in place is by alpha helix parts of the protein. You remember, the protein is a very complex structure. It's not just a simple structure. So you've got this complex back and forth of amino acid chains, and they've made these alpha helix turns. Those are hydrophobic whenever those turns take place. So you've got a bunch of those alpha helix all bunched together, and that creates a hydrophobic part of the protein. And then these big loops at the top and these big loops at the bottom, these are hydrophilic. So whenever that protein is put into that membrane when it's being made, it stays there because of the hydrophobic inside and the hydrophilic top and the hydrophilic bottom that like the environments that they're in. What are some of the functions of proteins? We're going to look at 6 of them real quickly. You can see these. These are also in your book. So the first is just transport. Those transmembrane proteins deal with transport, moving substances from one side of the membrane to the other, be it in or out. Enzymatic activity. Those membranes or those proteins that are in the membrane could cause reactions to take place inside or outside of the cell. And if it does, then those are enzymes. Cell surface receptors. So there's a signal up here that's detaching, and then it causes some kind of signal to happen down here inside the cell. The signal doesn't necessarily travel into the cell. That signal stops there on the outside of the protein and sends the signal to the cell itself then. Another is identity markers, carbohydrates, and the glycoprotein, protein connected to the carbohydrate top. And this is what you hear about when you hear about people talking about they need a match for an organ, they need a kidney donor match. They're talking about carbohydrate markers. And this is the way the cell interacts with itself. And your white blood cells are going throughout the cell looking at self, and they're feeling for these carbohydrate markers. And if they've got them, good. If they don't, they mark it as a bad cell. Let's eat it and get rid of it. Cell-to-cell adhesion. Proteins will stick to each other, and that will cause those cells then to be stuck together and not move. And then attachment to cytoskeletons and the extracellular matrix, holding everything together, tying everything together in the cell. Let's do some word association. Whenever you see membrane, what should be the response you have? It should be selectively permeable. That should be the thought that comes to your head. I don't have that typed in right here. Let's see. I think we can type that in. Selective. Oh. Okay, selectively permeable. That should be what you think about. What things are selected for? Well, if you're small, if you're polar, if you're uncharged molecules, like water, you'll pass through. And I know that's weird, but we'll even talk about some ways water passes through. They actually use aquaporins as well, more often than just passing straight through the membrane. And then if you're nonpolar, you pass easily, like steroids. If you're small and nonpolar, you'll pass right through that membrane, no problem. If you're small and polar and uncharged, you can get through there sometimes, but it's a little bit harder. What things are selected against? Well, large molecules, polar molecules, charged molecules don't get through very well. Glucose is a huge molecule, doesn't get through the cell membrane. Amino acids, huge molecules, don't get through the cell membrane very easily. So you have to look for molecules that are small enough if you're just going to pass straight through the cell membrane. Most, however, don't use that system. Most use a different system that we'll talk about in a minute. So if we're talking about water going through the cell membrane, primarily we're talking about it going through aquaporin. Aquaporins are new proteins that have been discovered. They're not new to the cell. They've been around for a long time for the cell, but they're new for us. We've just recently discovered them in recent science. So they are allowing water to pass through and other molecules. So primarily what we're looking at is the movement of water. And you can see that here. So water will move in the direction of high to low, passive transport. We'll talk about that word in just a second as well. And it will just go right through that membrane. Here you've got a model that we can create in the lab. You've got a membrane in the middle of this U here that is too big. The holes in this membrane are too small to allow the solute, in this case sugar, to go through. So water will pass through the membrane. And what happens is if you have a lot of solute on one side and very little on the other, then water will pass through that membrane and fill up the side with more solute. And the reason is because of free water. The solute that's in the water will cause the water to bind to different parts of it, and there won't be a lot of free water floating around. So if there's a lot of free water on one side of the membrane, not a lot over here, water will flow across that membrane in the direction of high to low free water molecules. So here's 3 examples that we look at: hypertonic, isotonic, and hypotonic. In a hypotonic solution, there is more free water outside, more solute inside the cell, so water flows in. In animal cells, it causes that cell to burst or lyse. In plant cells, it causes it to create turgid pressure, which is important. That's actually where a plant wants to be, turgid pressure. It makes them all tight, gets as maximum amount of water in there as possible. In isotonic, you actually get a free movement of water to both directions. In hypertonic, you've got more solute outside of the cell than inside of the cell. For instance, if you drink salt water, it would actually cause your cells to lose water or dehydrate because there's solute in the salt water more so than what's in your cells, and your cells would lose water then. So your cell shrivels. In plants, that's called plasmolyzed, and you can see the cell membranes pulling away from that cell wall, and usually this happens right before the cell dies in plants. So how are substances transported across the membranes? Well, I've already mentioned the term a couple times. It's passive transport and active transport. In passive transport, no energy is required. Things flow down their concentration gradient. It's kind of like riding a bike. If you start at the top of the hill and you ride to the bottom of the hill, it's not very hard to ride from top to bottom. You don't have to put in a bunch of energy. You just hold on tight, right? Move your legs out of the way, let the pedals flow freely, and every once in a while tap a brake or something, and you just easily go and coast all the way down the hill. If you're trying to go up the hill, though, if you're down here and you're trying to go up here, that's not as easy anymore. That requires energy. So that is called active transport. Energy is required to flow against the concentration gradient. You're going from an area that's not a high concentration towards an area of higher concentration. You have to put in some energy to make that go. Both of these use proteins. Now, in passive transport, you can just get simple diffusion to take place, which is no proteins. But a lot of passive transport that's happening does actually use a protein. It could use a channel like this. It could use some kind of open and close function that you see here. It could use some kind of gate. There's many examples there. We'll actually talk about some more in class. In active transport, you see here you have to add energy. Now this is the sodium-potassium pump. It's the one that's most used, and we call a lot of these pumps in cells, where you're bringing in a certain amount, you add energy, it will force those against their concentration gradient to the other side of the cell, and then it does the same thing in return the other direction. What are some other important mechanisms of transport? Well, those are called endo and exocytosis. In endo and exocytosis, you actually got the membrane pinching in around a substance outside and bringing it in as just one big chunk. If it's a food substance, then we call that phagocytosis. If it's a liquid substance like water, we would call that pinocytosis. And you can see those here, and it's just the cell just circles around it, brings it in. It's inside of a membrane now, inside of the cell. The last one here is a receptor-mediated endocytosis. This would have receptors on the outside of the cell, and whenever those receptors have a signal that attaches to them, it causes the cell to just take in massive amounts of stuff on the outside. Very cool as well. We'll look at this a little bit more in class too.